Welcome to this introductory lecture on cell biology. Today, we are going to explore the mitochondrion, which is universally known as the cellular power plant. We'll build our understanding from the ground up, looking at its unique physical architecture, the specific compartments it contains, and the mathematical principles that explain its incredible efficiency. By the end of this video, you will understand exactly how this microscopic structure keeps you alive and energized. To start our journey, I want you to imagine your body as a bustling, massive city that needs an enormous amount of electricity to keep its lights on and factories running. In the microscopic city of your cells, the mitochondrion is that central power plant. It is not just a simple, single-chambered battery, but a highly organized, multi-layered facility. Just like a real power plant has different rooms for fuel processing, turbines, and waste management, the mitochondrion uses internal walls to create distinct spaces. This brilliant design allows complex chemical reactions to happen simultaneously, without interfering with each other. Now that we have an intuitive picture of a power plant in our minds, let us look at the formal biological definition. In formal terms, a mitochondrion is a double membrane-bound organelle found in the cytoplasm of almost all eukaryotic cells. This double membrane architecture is not just a detail, it is fundamental to how the organelle functions. The overall structure is defined by four distinct regions: the outer membrane, the intermembrane space, the inner membrane, and the matrix. Each of these four regions plays a highly specific and critical role in energy production and cellular regulation. Let us break down these four distinct regions in more detail. First, the outer membrane acts as a porous skin that encloses the entire organelle, allowing small molecules and ions to pass freely in and out. Inside this is the inner membrane, which is highly folded into complex structures called cristae. This inner membrane strictly controls what enters and exits while housing the actual machinery for energy production. The space trapped between these two membranes is the intermembrane space. Finally, the dense fluid enclosed entirely by the inner membrane is the matrix, containing unique DNA and metabolic enzymes. Having discussed the four regions, let us visualize them. On your screen is a cross-section of a mitochondrion. Notice the smooth outer membrane enclosing the entire organelle. Just inside, you can see the intermembrane space. The most striking feature is the inner membrane, which folds inward repeatedly to form the cristae. Deep inside these folds lies the matrix, dotted with circular DNA and tiny ribosomes. This specific physical layout is exactly what allows the organelle to function as a highly efficient power plant. You might wonder why this specific, complex structure matters so much to your daily life and overall health. This unique double membrane architecture is the absolute key to cellular respiration. This is the vital process that generates adenosine triphosphate, or ATP, which is the primary energy currency of your cells. It powers your brain to think and your heart to beat. Furthermore, mitochondria play a critical role in deciding when a cell should naturally die, a protective process called apoptosis that helps prevent diseases like cancer. Malfunctions here lead to severe metabolic diseases. Building on the immense importance of the mitochondrion, we must truly appreciate the genius of its physical design, specifically the membrane folding. These internal folds, known as cristae, drastically increase the available surface area for energy production. By folding inward repeatedly, the inner membrane creates a massive internal workspace without requiring the overall organelle to grow any larger in size. This means the cell can pack significantly more energy-producing proteins into the exact same microscopic volume, maximizing efficiency. To truly understand the impact of this folding, we can measure this biological efficiency mathematically. We use a concept called the surface area amplification factor. The formula is A sub F equals S inner divided by S outer. Here, A sub F is a dimensionless number, telling us exactly how much larger the inner membrane is compared to the outer one. S inner represents the total surface area of the highly folded inner membrane, and S outer is the surface area of the smooth outer membrane. When the value of A sub F increases, it physically means the mitochondrion has more folds and can produce much more energy. Let us see this formula in action with a concrete example. Imagine a researcher is examining a human liver cell mitochondrion under an electron microscope. We are given that the smooth outer membrane has a surface area of 3.0 square micrometers, and the highly folded inner membrane has a total area of 15.0 square micrometers. We are asked to find the amplification factor. We write down our formula and substitute 15.0 for the inner area and 3.0 for the outer area. Dividing these gives us exactly 5.0. Physically, this means the inner membrane holds 5 times as many enzymes. Now that we have successfully calculated the amplification factor, let us connect it directly to the actual energy output of the cell. The inner mitochondrial membrane is densely packed with ATP synthase enzymes, which are the literal engines of energy creation. Therefore, the total energy production capacity of the mitochondrion depends entirely on the total surface area of this inner membrane. We can measure this production rate in units of ATP generated per second for every single square micrometer of that membrane. Understanding this link is crucial for our next calculation. This brings us to our next important mathematical relationship. To find the total energy capacity of the organelle, we use the formula total ATP equals S inner multiplied by P. Let us break down these symbols. Total ATP is the overall energy produced per second by the entire mitochondrion. S inner is the inner membrane surface area we discussed earlier, measured in square micrometers. The variable P represents the specific production rate, which is the number of ATP units produced per second on a single square micrometer. If the inner area increases, the total energy output increases proportionally. Let us apply this new formula to a heart muscle cell, which requires massive amounts of continuous energy to keep your heart beating. We are given an outer membrane area of 4.0 square micrometers, an amplification factor of 8.0, and a production rate of 1,000 units of ATP per second. First, we must find the inner surface area. We rearrange our amplification formula so that S inner equals AF times S outer. Multiplying 8.0 by 4.0 gives us a total inner membrane surface area of 32.0 square micrometers. Continuing with our heart cell example, we now have the inner surface area, which we found to be 32.0 square micrometers. We are asked to find the total ATP production capacity. We take our total ATP formula and substitute the known values. We multiply the inner area of 32.0 by the given production rate of 1,000. This gives us a final answer of 32,000 units of ATP per second. This beautifully demonstrates how highly dense cristae allow heart cells to generate massive amounts of energy in a tiny microscopic space. Having successfully calculated the energy output for the heart cell, let us formally prove the mathematical relationship between membrane folding and total energy capacity. We want to establish exactly how the amplification factor scales the energy output. We start our proof with the base energy formula, where total ATP equals S inner times P. Next, we recall our definition of the amplification factor. By isolating the inner surface area algebraically, we establish that S inner is exactly equal to the amplification factor multiplied by the outer surface area. This sets up our substitution for the next step. Now we will complete our proof by combining these ideas. We take the expression for S inner that we just isolated and substitute it back into our original energy formula. This gives us total ATP equals AF times S outer, all multiplied by P. By rearranging the terms, we clearly see that the total energy output is directly proportional to the amplification factor. We have mathematically proven that simply by increasing the folds of the cristae, the mitochondrion linearly scales up its energy production without needing to grow any larger in overall size. We have covered a lot of ground today, so let us summarize the key takeaways. First, the mitochondrion is a masterpiece of biological engineering, utilizing a double membrane system to create distinct environments for cellular respiration. Second, the extensive folding of the inner membrane into cristae is crucial as it maximizes the surface area. We learned how to quantify this using the surface area amplification factor. Finally, we proved that this folded architecture is the exact reason your cells can generate the massive amounts of energy required for you to live, breathe, and learn.